Greetings of the day. Myself Sharviri Jitin Kulkarni, and I am going to present on the topic of DNA repair mechanisms, of which we will be focusing first on light repair and second on SOS repair. So we'll begin with the introduction. DNA, like any other molecule, can undergo a variety of chemical reactions because DNA uniquely serves as a permanent copy of the cell genome. However, changes in its structures are of much greater consequence than are alterations in other cell components such as RNAs or proteins. Mutation can result from the incorporation of incorrect bases during DNA replications. As we all know, DNA comprises of bases adenine, guanine, cytosine, and thymine. In addition, various chemical reactions and changes occur in DNA either spontaneously or non-spontaneously or as a result of exposure to chemicals or radiations. Such damage to DNA can block replication or transcription process, which are quite essential for all living organisms cell uptake and can result in a high frequency mutation consequences that are unacceptable from standpoint of cell reproduction. To maintain the integrity of their genomes, cells have therefore had to evolve mechanisms to repair damaged DNA. These mechanisms of DNA repair can be divided into two general classes. First will be the direct reversal of chemical reactions responsible for DNA damage, and second will be the removal of damaged bases followed by their replacement with newly synthesized DNA. Where DNA repair fails, additional mechanisms have evolved to enable cells to cope with the damage. As mentioned in the picture beside, DNA molecule or the two strands of DNA has been mentioned, one comprising of 5' end and another of 3' end. Here it is described about the bases as we have talked about of guanine, cytosine, thymine, adenine, which are linked with sugar phosphate backbone, and nitrogenous bases, as mentioned, which have major and minor groups. So now we'll begin first with the light repair mechanism. This mechanism is also known as photoreactivation, which is a DNA repair mechanism that uses light energy to fix UV damage to the DNA. This damage is usually in the form of pyridine dimers, which are abnormal covalent linkages between two adjacent pyrimidine bases. So as we can see in the image described, we can see the photoreactivation repair, which is caused which is caused by the TT, that is TT linkers having double bond which are covalently linked, which are also known as pyrimidine dimers, and the adjacent which are adjacent to each other, and the bombardment of UV light towards it. So during this photoreactivation, an enzyme called DNA photolyze. So we can see in the image the photolyzed enzyme is released, which then binds to the dimers and uses light energy in the wavelength of 310 to 500 nanometer to break the cross linkers formed between the dimers. This restores original pyrimidine bases which are formed or which are seen in the normal double-stranded DNA molecule, and the damaged DNA gets break. Photoreactivation is a type of direct reversal repair, which means it fixes DNA damage without needing to replace or excise it. So we'll begin with the characteristics of this repair mechanism. First, we'll be focusing on specificity. Photoreactivation is highly specific to UV-induced pyrimidine dimers, making it a targeted repair mechanism. So here, the key aspect of this mechanism is photoreactivation is a highly specific mechanism. So this mechanism will only occur at the targeted site, which highlights itself as a targeted repair mechanism. Second will be the second characteristic will be the light dependency. The repair process requires visible light to activate photolyze, which is a type of enzyme used here, differentiating it from other DNA repair mechanisms that operate independently of light. Third will be presence across species. Photoreactivation is found in many organisms, including bacteria, fungi, plants, and some animals. However, it is absent in placental mammals, including humans. So, this third point explains or highlights the fact that photoreactivation or the type of light repair mechanism is generally found in the prokaryotic genera rather than eukaryotes. So, we can see the inclusion of bacterial species, fungal species, plants, which includes the domain of prokaryotes and very few animals. So Eukaryotic genera lacks this repair mechanism. Further, we'll be processing with the importance of this mechanism. Photoreactivation provides an efficient way to directly reverse UV damage induced, protecting cells from mutagenic and cytotoxic effects of UV radiation. This mechanism is especially important for organisms that are frequently exposed to sunlight as it helps to maintain their genomic integrity and cellular functions. So we can relate it to the fact that many microorganisms, plants, fungi, and any other species of every prokaryotic genera are always exposed to sunlight. So chances of this genera to get exposed to such UV damage in their DNA or cytotoxic effect in their cells, which ultimately leads to this damage, can be can be reversed by this type of light repair mechanism, uh, which is proceeded with the inclusion of photolyzed enzyme. So now we'll be focusing on the second repair mechanism, which is SOS repair. It should be noted that this SOS repair mechanism occurs only in prokaryotic genera. The SOS response is a global regulatory network in bacteria that is activated in response to the DNA damage. So as the first part highlights itself as this DNA repair occurs only in prokaryotes, we can see that the organisms associated with this repair will only be the bacterial species. While it's primarily associated with prokaryotes, here are few reasons why it is not typically found in eukaryotic genera. First will be the complexity of eukaryotic systems. As we all know that there is a diverse array between the prokaryotic cell typically and a eukaryotic cell typically. Eukaryotes have more complex and diverse DNA repair mechanisms, so they don't need any usual type of SOS repair mechanisms in their cell machinery. They possess multiple specialized pathways, or we can also say biochemical pathways, which are induced in their cells for repairing different types of DNA damages such as nucleotide excision repair, base excision repair, mismatch repair, and homologous recombination. This complexity reduces the need for single broad spectrum response like SOS system. Second point will be highlighting of genomic organization. Prokaryotic genomes are typically simpler and more streamlined compared to the eukaryotic genomes. So we all know the basic fundamental concept that prokaryotes have evolved way earlier than eukaryotic cells. Hence, the SOS response is well suited to the more straightforward genomic contents of prokaryotes only, where rapid and coordinated responses to DNA damage are crucial for their survival. Third point will be highlighting on regulatory mechanisms. Eukaryotes have evolved sophisticated cell cycles, checkpoints, and signaling pathways that tightly regulate DNA repair process and cell division. We all know that eukaryotic organisms have a correct signaling between their cell cycles and between the associated cells of any pathways. These checkpoints allow cells to pause the cycle and repair DNA damage before proceeding. So cell gets signal accordingly. The checkpoints allows the cell to pause or to stop the replication process. So the DNA repair can be proceeded, reducing the necessity for a system like SOS. So now we'll be highlighting on the fourth point of translation synthesis. The SOS response involves a error-prone translation synthesis where specialized DNA polymerases replicate over damaged DNA. Eukaryotes also have translation synthesis synthesis polymerases, but they are regulated differently and integrated into a broader DNA damage response pathways. Fifth point will be highlighting on the evolutionary divergence. The evolutionary pathways of prokaryotes and eukaryotes have diverged significantly. We all know this. This is the fundamental difference between a typical prokaryotic genera and a eukaryotic genera. The SOS response likely evolved specifically in prokaryotes due to their particular environmental pressures and life cycles. So we can see that the typical prokaryotic cells, which are primitive, they typically lack the property of handling the environmental stress and the life cycles which are occurring intracellularly in their cells. Overall, while eukaryotes do have mechanisms to deal with DNA damage, these are typically more specialized and integrated into complex cellular regulatory networks, making them broad spectrum SOS response unnecessary. Now we'll begin with the characteristics of this mechanism. The core mechanism will be as follows. The SOS response is a global regulatory mechanism used by bacteria to address severe DNA damage. Here's a detailed look at how it works. So, the image described explains us about the double stranded DNA base pairs having end processing to generate single stranded DNA. As we can see in the step B, then further we can see polymerization occurring and activation of RAK. Now this RAK is a type of protein which typically gets attached to the 3' end of the DNA molecule. Further, RAK filament interacts with the Lexay repressor, activating it for self-cleavage. Now, here the inclusion of Lexay, which acts as a repressor, also begins, which has a typical role of activating itself for self-cleavage in the single-stranded DNA molecule. As we can see that the RAK has attached to the 3' end of the DNA molecule. Further, the Lexay molecule gets attached to the single stranded DNA and the SOS genes get repressed. The Lexay autocleavage happens and transcription of SOS genes begins. Then it leads to the disruption of Lexay, which acts as a repressor. RNA polymerase binds and SOS genes get induced. We'll begin first with the induction trigger. The SOS response is initiated when DNA damage occurs, as we all know, such as from UV radiation, chemicals, or any other stressors that cause DNA lesion, that is, breakage of the DNA strands. Second point highlights about the role of RAK protein as they have seen in the image. First will be its activation. Single-stranded DNA molecule, that is ssDNA, accumulates at the site of the damage, leading to the activation of the RAK protein. We'll see its function. RAK binds to the ssDNA, which is single-stranded DNA, and forms a nucleoprotein filament. That is the filament we have seen in the images, which is attached to the 3' end of the single-stranded DNA, which facilitates the cleavage of the Lexay repressor. Third point highlights about the role of Lexay protein. Lexay protein acts as a repressor. Under normal conditions, Lexay represses the expression of SOS genes by binding them to their operators. It also helps in cleavage. Activated RAK promotes the self-cleavage of Lexay. So we have seen in the image that the role of Lexay repressor is typically in acting as a cleavage molecule, reducing its level and thereby de-repressing the SOS genes. Fourth will be the SOS genes. The de-repression of Lexay leads to the expression of over 40 SOS genes involved in DNA repair, replication, and cell cycle regulation. These genes include DNA repair enzymes such as UVRA, UVRB, and UVRD, which are involved in nucleotide excision repair. Then, translation synthesis polymerases. These are a type of enzymes such as Pol4-TeInB and Pol5-UMUDC that can replicate over damaged DNA, which we have seen in the image. Further, cell division inhibitors such as SULA, which inhibit cell division to allow time for repair. Now, these cell division inhibitors act as a key point in this mechanism, as we all know that when the process of DNA repair mechanism gets activated, it is necessary to give signal to the cell and the pathways associated or the pathways which are working intracellularly in the cell to stop the repair mechanism or to stop the effects or to stop the biochemical effects which are undergoing in the cells. We'll begin with the fifth point of translation synthesis. The process allows DNA replication to continue past damaged sites, albeit in an error-prone manner. This can introduce mutations but ensure survival under conditions where DNA is heavily damaged. Now, this point stresses upon the translation synthesis process, wherein we can see that mutation can occur in the cell when it is undergoing SOS repair, but at least it is ensured that the cell or the DNA strands which are undergoing this type of mutations can get some chance of survival when it is heavily damaged. So we begin with the sixth point of regulation of the cell cycle. The SOS response includes mechanisms to halt the cell cycle, preventing the propagation of damaged DNA and providing time for repair. We can see in the image described beside showing the normal DNA strand, SOS box which is involved, SOS switched off part of the DNA, damaged DNA which gets repaired, and at the site at which the SOS response gets switched on. We'll begin with the stages of the SOS response. First will be the initiation. DNA damage results in the accumulation of ssDNA, which is single-stranded DNA, which activates RACE. Second point of regulation. Activated RACE facilitates the autocleavage of LXA, lifting repression on SOS genes. Third point is of response. SOS genes are expressed, leading to increased DNA repair activity, error-prone DNA synthesis, and cell cycle arrest. Fourth will be the recovery phase. Once the DNA damage is repaired, the levels of single-stranded DNA, ssDNA, decrease, inactivating the RACE protein. Lexay reaccumulates, repressing the SOS genes again and allowing the cells to resume normal functions. The SOS response is crucial for bacterial survival in environments where DNA damage is very frequent. It balances need to repair DNA with the risk of introducing mutations which can be beneficial or deleterious. We'll be focusing lastly on the bibliography which I have mentioned of both images as well as content. So we can see the content from which I have taken the concepts mentioned and the images. Thank you.